So my title is a development of a implantable device for continuous monitoring of blood biomarkers. So this is an overview of this presentation. So firstly, we will take a look at the overview of what is optical fiber sensing technicals. And then we'll talk about project, aim and objectives, and some work packages and milestones. Also, of course, the time plan. And also some interesting initial results. We will talk about a bit of sensor fabrication results and functionalization, also the measurements, and then talk about the future work. So so you all know about a traditional method of antibody measurement. So this here is showing the traditional ELISA method to detect the target antibodies. So the visible enzyme reaction will indicate the concentration of the target antibody, for example, IgG. So which means like the process of that ELISA method is a visible response, and also higher sensitivity and specificity. Also, however, it has so many disadvantages. So expensive laboratory equipment and the cultural medium required also. Also the bulky equipment and non-reusable and so on. So optic fibers. So I think most of people know the optic fibers because of its optical communication uses, right? But optic fiber also can act as a platform of sensing system because of its lightweight, like hair size, and a small dimension. So typically, a typical diameter of a single-mode optic fiber is around 125 micrometers. And also, it's immune to the electromagnetic interference, which means it can safe use in the hazardous environments. So that corner is an SEM image. We take it on top of the optic fibers, as you can see how small it is. So working principle of the optic fiber sensor system is fairly straightforward. So we got light source, optic fiber, and sensing elements is part of optic fiber, and then optic detector. It can be photodiode or spectrometers. So the light will emitted from light source and propagating through the whole optic fiber. And the sensing element, when they measure it interacting with the sensing element, so there is a optical signal changes. So this photodetector or spectrometer gonna capture this optical change. Then we can indicate the measure rate. Particularly for the biochemical optical fiber sensor system, the functionalization is a key step for fabricating the sensors. For example, here is a tip of fiber. We coat the functional film on there. In order to improve the sensitivity and selectivity of the specific measure rate. For example, here we try to capture the pink circle one. So we have to coat the surface of the fiber, the sensing element with specific functional film. So there is several methods to let us to generate this functional film, layer by layer absorption, and molecular imprinting, and surface plasma renaissance, also metal organic frameworks. I would like to introduce this sensing platform, optical fiber sensing platform here, as it's gonna used in this project. So which is long paper grating. So typically we have a single optical fiber, jacket, cladding, and core. So if we have a periodical grating structure here in the middle of the core of the fiber, And then we launch a fundamental mode light, which is like on the spectrum, we can see it's a flat of the transmission spectrum. When the light passes through the Lampere grating area, so after that, we will have several attenuation bands. So these bands are very sensitive to the surrounding area, which is a refresh index. So then we can use that to detect the surrounding environment. Okay, so then we talk about this project. So the aim of this previous project is to develop a reversible continuous blood monitoring optical fiber sensor. And also this allows us to provide a proof of concept that implantable monitoring is achievable. As you can see, the image showed here, the final aim of the middle term goal of this project is kind of like embedding the optical fiber in the tissue or the vessel, and then we can detect the different biomarkers, for example, the antibody, in real time, and we have a smart device interface and allows us to like transform the optical data to the digital and send by wireless to the terminal, for example, the PC or laptop. And the objectives can be is for main part. So investigation and fabrication of the grating-based optical fiber sensing platform. And other will be development of initial functional coating. For example, for the IgG, particularly, is a IgG binding peptide. And then we characterization of the sensor's sensitivity, reversibility, and repeatability to the targeting measures. So here is the IgG antibodies. And also develop a test kit with tissue sample and try to refinement of the coating based on above objectives in the blood sample. So we got 4 work packages. So package 1 is sensor development and fabrication process. Work package 2, sensor characterization and evaluation. Package 3, blood test and or phantom measurement. And package 4 is next stage development, which is kind of potential journal publication or some further grant application. So we got 4 deliverables and 2 main milestones. So milestone 1 is month 9. So we aim to have 5 sensors successfully tested in the non-blood environment with a acceptable limit of detection and reusability. Here it can be used 3 times to measure the IgG. And milestone 2 is month 12. So 3 sensors successfully tested in the blood. So this is a time plan of the whole project. So the 4 deliverables and the milestones, which is after the Q2. So now we are here, just start the project. All right, so yeah, that will be some interesting part about the initial result we got to support this project. So firstly is LPG fabrication. So this slide shows how we fabricate the non-pair grating in our research lab. So we have an optical fiber here. It's sitting on the V-grow fiber holder, and the laser shines through the lens and phase and amplitude mask and strike on the fiber core. And you can see these figures with the time and with length of LPG. There is 2 attenuation bands appear. So once we got that spectrum, which means it's able to measure the surrounding refresh index. Also, we apart from after we have the non-pair grating, then we also like built this simple microfluidic setup, which is for mimicking the in vivo situation and also avoiding the measured sediment because we are flowing the sample solution all the time. So here we homemade some like 3D printed holder to molding, demolding of the part of flow cells. So you can see the lumpy grating is sitting between the flow cell lid and bottom, and then we use the holder to make it tight. And during the lumpy grating area, we got inlet and outlet tubing, which allows us to pump in the sample solution. And also in the meantime, we can measure the transmission spectrum in real time. So same setup, we tested the bio-LPG for the refresh index monitoring. And so here we pump in different solution with different refreshing index value. And figure B and C is the spectrum change and also dynamic change of the alternation band. Based on that figure, you can see how quick response of the different refresh index. And also the figure D shows the calibration curve. Then we can see like for the bio-LPG, for the refresh index sensitivity, we can reach to 335 nanometers. So once we test that, then we can go to the functionalization step. So for the specific sensing to IgG, so firstly, after the hydrolyzation and silicon nanoparticle and gold nanoparticle, then we will have the golden shared silicon nanoparticle, which allows us to bind the IgG binding peptide. After that, the sensor is ready to detect the IgG antibody. This slide shows we are able to like synthesize the golden nanoparticle in the lab. From the SEM imaging, you can see how uniform morphology of the golden nanoparticles. And also the typical size of the particle is around 20 nanometers. And then sensor fabrication. So this is a transmission spectrum recorded during the sensor fabrication. The expanded band is shifted here, which indicate the successfully coating. And also from the SEM imaging, you can see the gold shared nanoparticles are coated successfully on the optic fiber surface as well. So this is the initial dynamic monitoring using the microfluidic setup. At the first beginning, we just pump in the PBS solution in the flow cell, and you can see there's like a flat, so nothing response, which is good. And then we add the IgG into the flow cell, and we can see a very clear association process. We did twice of the experiment. We can see clearly in both circle, we can see that there is roughly 1 nanometers change due to the same concentration of IgG. Okay, so yeah, so for the next, we'll keep investigate the reversibility. Also, we try to increase the sensitivity from the basic sensor platform. And also, we will add blood flow phantom and try to like drafting potential publication based on the data we got. So yeah, now you understand optical fibers. So optical fibers can fit so many applications. For example, the drug detection, and for example, today's immunosensing, we can detect antigen, antibody, Also, a portable pyramid monitoring kind of medical device. Also, some biomarkers like CO2, ammonia also can be detected by optical fiber sensing system. So the potential collaborating can fit the gap between the clinical challenging and the technical limitation, and we will make people's life easier. All right, that's all for my presentation. Thank you.